Well, good afternoon, everybody. Welcome to Marloes' inaugural professorial lecture in the Department of Chemical Engineering. My name is Philip Martin. I'm Head of Department of Chemical Engineering at the moment. I've got a few words of introduction to Marloes before she starts. Well, Marloes is from Roosendaal, a small city in south of the Netherlands. And she obtained a Master's in Chemical Engineering from Eindhoven University of Technology in the Netherlands, specialising in polymers and composite materials. And after that, she went to do a PhD with Professor Patrick Wagner at the Hasselt University in Belgium, where she developed polymer-based sensors for detection of neurotransmitters. And after that, in 2014, she moved to the UK, initially at Queen Mary College for postdoctoral work, but soon started an independent career as a lecturer at the Manchester Metropolitan University, just down the road. And then from MMU, she moved back to chemical engineering at the University of Newcastle and was promoted to chair there in bio-inspired materials back in 2023. And then later that year, Marloes transferred to the University of Manchester to the Department of Chemical Engineering in a new chair in engineering biology, which aligns with interests in biomimetic materials. So Marloes has published over 90 papers on molecular imprinted polymers and has been granted sort of 4 patents on novel sensor platforms. And she's also a very keen sort of science communicator. Has won various awards for being a role model for women in STEM subjects. So 2024, well, last year, it's been a good year for Marloes with the birth of Neil. He's looking forward to the lecture, wants to be finished as quickly as possible. Neil was here and also awarded the RSC Macro Group UK Early Career Award, given to a UK-based polymer scientist who shows great promise for the future. So I'll hand over to Marloes to start her lecture.
Okay, well, thanks very much, Phil. So nice to see so many of you here, and thanks for coming to this inaugural lecture. Here I'm just going to give a snapshot of the research we've been doing. So hopefully I've pitched it to everyone so can kind of follow what we've been doing, showing the latest developments. So if I look in the room, all of you are very advanced biological sensors. So let's see if this starts. You have 10,000 taste buds. When it comes to smell, you can even smell trillions of things. But what's more important, what you have in your body as soon as you have foreign invaders, you will have a response in terms of an antibody. So antibodies we know now because of COVID-19, they're very good at very selectively and specifically recognizing certain things that enter your body. So that's these antibodies. So what we're doing is we're looking at materials that are kind of inspired by this selective recognition. And you can tell that Neil already had enough of this. But in terms of healthcare applications, I think we can't live without them anymore. So this has gone from the 1960s, where they first started developing tests for glucose, the pregnancy tests. Obviously, we know the lateral flow test for COVID. And recently, also, if you look at in terms of cancer treatments, where they are using monoclonal antibodies, this has really transformed how we treat cancer and other chronic disorders. Now, if you look at the list of best-selling drugs, so besides the ones that are mRNA vaccinations or the ones that are weight loss at the moment, these monoclonal antibodies, of which there are like around 500 in the market, they make up the majority of these top-selling drugs. However, I will show to you why I don't think that's sustainable. I think they're expensive, not very stable, and obviously I will also give you a solution to this, and that's what our research is about around the development of polymers. Now, if you look at your biological sensors, obviously they are highly effective. It's been over millions and millions of years of evolution, so it's very good at actually detecting certain things. But it's also very specifically designed to you. So imagine if you have a very specific enzyme, for instance, in your stomach, which is acidic. It works under very specific conditions. It obviously likes your body temperature, and it likes a very specific pH. So that is a bit of a problem. They can also trigger allergic reactions because they are biological material. And if we need to produce them, we're obviously not going to extract these antibodies from us. We're going to extract them either what people traditionally used to do from animals, or now we also have recombinant technologies. But all of these technologies, they are quite expensive, and there's still a lot of hidden animal use around the use of these antibodies. For instance, if you look at diagnostic testing, you can find papers saying that we still use around 1 million animals per year in the European Union, so not in the world, that are used for the production of these antibodies. And you can imagine they are used, but most likely they are being sacrificed as well. Now, what's the solution? So here you can see, hopefully you recognize this COVID test. Now, if we look at this, so this was kind of, I guess, a trick invented by school children. This is not a positive test. Well, it is a positive test, but actually people didn't add something related to COVID, but you can have vodka, brandy, Red Bull, whatever you added to it. And this kind of underlines this effectiveness of these antibodies, but essentially what is happening, you have something that changes the pH, and as soon as you do that, your test is confused. Because it has these antibodies that work under these very specific conditions in your body, and it's not able to capture that well or to bind or to deal with these changes. So that's why most of the time you actually use buffered solutions. Now, what's the solution to this? So I mentioned it, my title is an engineer in biology, so hopefully at the end of the talk you have a rough idea of what that's like. So we're inspired by biology to engineer new materials. So design synthetic materials which should overcome all of these challenges by being low cost, versatile, and cost effective. Now, and the obvious answer to it is I am a polymer scientist, so I say that the answer is polymers, even though there are probably more solutions out there. In terms of the people here who are non-scientists in the audience, if you think of the word polymers, think of it as like a long bead, something which is high molecular weight, which is made up out of the same repeating units. So you probably know this from your plastic bags, which is a polymer as well. So you might better know it as plastics, which is not 100% the same. But these materials, these polymers, can do all of the things that antibodies can do while they have these advantages. Now, when it comes to the road in terms of my research development, so as Phil mentioned, I did my master's in Eindhoven University of Technology, where I specialized in what we call polymers and composites, working with Gijs and Professor Cor Koning, where we were making very defined polymers. So in that sense, there was no real practical application yet. It just meant that we had to make very, very specific structures. And the more precise, the better. So this involved an 8-step synthesis procedure, which if people who've seen me in the lab know that being very precise was not my strong suit. Because imagine if you fail somewhere and along these 8 steps, you have to go all the way back to the beginning. But what I did really enjoy about this and what they picked up on is I really liked the practical application of this. So I was making materials, so this is essentially a type of polypeptide, so very similar to proteins. This kind of shows that it had a very well-defined molecular weight. And you can use these materials because they mimic the proteins in your body to start influencing certain processes. So in this case, we look at calcium carbonate crystallization, but they can also be used for drug delivery. So I did like the fact that we made these materials, but what we were going to do with it, I thought was quite interesting. So moving away from the 8-step synthesis procedure, and then I did my research internship in Brazil. So this was kind of like the city where I was. Well, not exactly the city where the company was, but I'm not sure what happened. There must be someone in the Zoom meeting. So this is, I think it has one of the best beaches in Brazil, they say, so it's like around here. So during this 4-month internship, I think someone might need to mute themselves. Okay, we'll figure it out. Someone in the Zoom meeting might be able to do that for you. Anyhow, as I did my 4-month research internship, I had a bit of time to think about what I wanted to do. And that's how I ended up doing a PhD in Belgium with Professor Patrick Wagner, because there we had polymers, so that was kind of building on towards my master's, but it had a very real application working with a hospital. So the focus was much more like on the multidisciplinary element, which you still very much see in my research, and the clinical application. So this is Professor Patrick Wagner, who I did my PhD with. And I remember when I was speaking to him over the phone, my interview actually, because it was in Brazil, the internet connection wasn't very good. So it actually happened to be a phone interview, even that got cut short. But he introduced the concept of molecular imprinting to me. And for my students, I used to kind of give this little video, but I realized Jurassic Park is from 1993, and a lot of our students are much younger now. But the concept of molecular imprinting is actually very similar to biological imprinting. So, and this is like a quote from the movie. So, in essence, what it means, so something hatches from its egg, or imagine a baby, or whatever it sees first, it's kind of imprinted by it. And that's kind of like, that obviously has the biological function, that whatever they see around them, they're quite comfortable with it, and they like that environment. Now, if you think of molecules, they have this type of molecular memory as well. Whatever they're in contact with in the beginning, and I will explain what that's based on, they preferentially like in the end, more or less. And this is what this concept of molecular imprinting comes back to. So how kind of it works? So in essence, these are porous materials with high affinity binding sites. And imagine of it a bit like being like a Swiss cheese. So you have all these binding sites in there, and I will show kind of how this works. So imagine you want to detect something. So this is what we call our target. We want to detect it. We have monomers that kind of interact with it, which are the building blocks of your polymers. We build a network around it, and then we extract the template that we had in the beginning. So what you end up with, if you look at an enzyme, this is pretty similar to an enzyme. You have an active site, so you have like some chemical functionalities here, and then you have like a robust 3D structure around it. So in essence, very different in terms of what it will look like, the chemistry or the structure, but what we are mimicking is these type of interactions that you've got, so the non-covalent interactions, so for instance ionic interactions, hydrophobic interactions, hydrogen bonds, plus then that you have an impact of these cavities have a very specific size and shape. So all of that together leads to the fact that we have very specific and selective recognition. Now, you don't need to imprint necessarily with the template that you're working with, and you will understand later on why that's important if we're working with, for instance, bacteria or viruses. So you can work with something that represents that. Nowadays, the non-covalent approach is probably the most popular option because it makes it much easier to extract your targets afterwards. And before, people used to market this as plastic antibodies or polymer antibodies because that's what people are more familiar with. But hopefully I will show to you that I think we're not doing the material justice if we're saying these are just replacing antibodies. They have many more functionalities that we can use, and this versatility of the polymers I think is what's really selling it. And to give you an idea, so normally the problem, I guess, when you work with a small molecule, these binding sites are really too small to visualize. If you work with something like this is E. coli, which we all have a lot of in our gut, so you can see this binding site is literally just an imprint of that. So it's basically essentially a hole with exactly the same size and the shape of the E. coli, just to give people a rough idea of what this would look like in practice. Now, I wanted to show you, so the idea of my PhD was to make catheters, and the reason why we're using these polymers is because they can withstand extremes of pH. And we wanted to measure in the bowels, and hopefully you can imagine this catheter had to go through the nose, so it had to go through the stomach acid, was exposed to extremes of pH, lots of enzymes. So if you would just have a normal antibody, it would be degraded long before it even gets to the bowels. So I was looking at monitoring of these neurotransmitters. And these neurotransmitters are really important because for a lot of bowel diseases, we don't really know what's happening in the bowels. People know it, they can take a sample through this catheter, which has to be sent out to the lab. But actually, if you want to monitor what's going on in vivo, you can get a lot of insight into a lot of functional bowel disorders. Because, for instance, with people with IBS, it's been postulated that they have more serotonin receptors, which is associated with pain in your gut. So that was the underlying idea of my PhD. This is nearly 10 years later. But we did get some kind of prototype out of it, and this is what I'm still working on with my PhD supervisor. So this is just to give you a rough idea. So we have what vaguely resembles a catheter. We have like a little lid on here, which is the engineering end, so it can actually pass through your bowels where your sensor is protected. Then this is not actual bowel fluid, but something that simulates it, and it's green just so you can get a feel for that it's bowel fluid. We would take up something there, and then you would use the electrodes which are already on the catheter to get a very quick electronic reading. With the idea is that it's actually being tested on patients now because it's basically similar to a normal catheter, and hopefully will provide some insight in people with bowel disorders, because now at the moment you're mainly treating the symptoms that they have rather than actually what's causing the underlying problems. But as you can tell, so I started my PhD, and this is nearly 10 years later, so there is a very, very long line to actually getting it remotely close to how it works with patients. Now, what I did do during my PhD, so besides just the development of these molecularly imprinted polymers, as an engineer, I also started looking at how we can use these materials in the sensors. So the way we did that is, imagine that all of us, but also heat and electricity, they always find the path of the least resistance. So imagine if we have something like this, where there's holes in there, heat and electricity will always pass through what's the easiest, similar to what we do. So this is kind of representative of what these molecular imprinted polymers are, and I think my dad made this schematic. This is a long, long, long time ago, because I wasn't very good with all that software then. You get some binding to it, and what you then see is that these pathways, or these pores, they are blocked. So what we postulated, what we call the pore blocking model, you have these materials which can very selectively capture these things, and they block electricity, but also heat going through. So in essence, they are increasing the resistance at the surface, and resistance both from electrical resistance, which people already knew and which they measured, but also they looked at the thermal resistance, and that's something that hadn't been reported before. So this is Bart, who I worked with during my PhD, and he designed a clever device. So this is kind of roughly how it should work. We have our materials here, and you can see you get very selective binding to whatever you want, and then you see that there's an increase in thermal resistance as soon as you get more binding. So you combine the selectivity of the polymer material with actually the ease of the readout of basically what is a temperature-based sensor. So he applied this to DNA, and then we thought at the time, why not try it with these materials? And actually this happened to work very well, up to the point where you can find over 50 papers basically using these molecular imprinted polymers, if not more, using these thermal resistance. So this was the thermal device that we then had at the lab, which are basically rebuilt later on. So this is kind of to give you a rough idea. This is what it looks like from the inside, in case you're interested. Very basic electronics, which is not my strong suit, but I'll talk about later how we worked on this. This is roughly the dimension. It was about 5 kilograms. At the time, it took around 1,500 pounds to build, but I'm sure, like, given inflation, it would have been a lot more expensive now. And this is part of what we had some of these patents on, were on using these molecularly imprinted polymers in combination with this thermal readouts. Now, how kind of I came onto this path of doing an academic journey. I initially was doing a postdoctoral research position in Queen Mary in London. So I was in Belgium at the time, where I met with Irene and Patricia, who Patricia was one of my colleagues, and then Irene already made the jump to the UK and was kind of really pitching this to me. So prepare me for my interview. So initially I moved to Queen Mary, and then fairly soon after that I got my first lectureship just across the road in Manchester Metropolitan University. So going there, these were the first polymers that I made. These polymers are, if I look at it from a polymer science perspective, they're not very pretty. So essentially these look like white, very coarse white powders. Whereas now we work with much more sophisticated nanoparticle systems. So in essence, I remember I had to transport this, and it very much looks a little bit like suspicious drugs when you look at it. And if you look under the microscope, this is what this looks like. So some people here will definitely recognize that this is not the way to go. But the first polymers I ever made that work, and then we started integrating them into these screen-printed electrodes. So this is where I first met Craig Banks. I gave like a talk, which you do when you're new, and you say, this is what I can do, and you pitch it, and he was like, oh, fantastic, we have all these screen-printed electrodes. Why don't you take your polymers and put them in there and start detecting things? And this is kind of how this first all started happening, and that worked out pretty well, actually, because with screen printing, if you know how that works, you can print sheets and sheets and sheets of materials. So almost like kind of an endless supply, which is far more than what I could measure at the time. Now, what came next? Kai is actually here in the room, so my first PhD student. Didn't know he was coming, so it's a surprise for me too, but he was already in the talk before that. So what we actually did is he rebuilt that setup, the one that we had during my PhD. It's still up and running, so it's still being used, so that's a while ago, so a very robust system. And for a while, as when you start your academic journey, not a lot of things materialized from this. So we had these screen-printed electrodes, but then we started to focus on two very specific applications. So actually, I realized if you want to sell these sensors, you need to look at where the need for it is. So after, I think, probably 2 years of nothing, we realized where like the two, I guess, key areas would be. Well, one was on antimicrobial resistance, which I got my new investigator award on. And then the other one on cardiac biomarker detection via NC3Rs. And I got these 2 grants in 1 week, so surely it was a good start, but I also remember a lot of discussions. Nick is also here in the room, a lot of discussions around all the comments that I got around the rebuttals I had to write for my new investigator award. It was a very hard journey towards getting there. But what it allowed me to do is to actually build my research group. So you need grant income to get people in. Nice picture of Ollie, isn't it? So the second PhD student, and we had Alex and Bobby, so slowly we started to get some postdoctoral researchers coming in who have a bit more experience. And that's kind of how this all started to go up and running. Now, I think when you start your academic career, obviously you don't want to do exactly what your PhD supervisor does. So you kind of need to find your own niche. And I was trying to work out for myself kind of what made that transition to what I was doing differently. And I think the key determining factor there was we had these microparticles, which are very heterogeneous, didn't work that well at all. So we went from micro to nano, essentially. And this is where we first started working with Francesco Canferotta. Now it's now called Trozaro. And they had developed an approach, solid phase synthesis, which what we're still using, where you can actually make these material nano-sized. So what do? So hopefully you can see, compared to what we had before, which is very heterogeneous, very big bulky particles, we went from something much smaller, much more homogeneous, but also being water soluble, which made a big difference. And what we claim is also biocompatible, so you can start using them for other applications. And I think this is actually the turning point, because suddenly these materials are much more easier to functionalize, because you can just deposit them onto layers, and they start to actually compete with antibodies. Because before, the affinity was always a problem. Now, so there we can do this direct competition with immunoassays, and also we can directly functionalize them on the thermocouple. So this kind of looks a bit like this. And then some of the work that we started to do is we took the same setup, but rather than having these very like broad particles, which didn't stick well to the surface, would come loose, weren't very selective, we can actually have multiple thermocouples here where we can have one target. Hopefully you can see here that there's an increase of a line, which actually means when you see this increase in thermal resistance actually refers to the fact that you get binding to your polymer material. So this was for emerging biomarker. And then we could have 2 control systems at the same time. So it started to get a lot more applied in the sense that, and we start to get limits of detection which are more similar to what you would need for biological applications. But also we can start to integrate controls into our system so we can measure much more. Now, after all of this, what you get is you start building your research group. Another lovely picture. Luckily, there's still quite a few people here in the room. Kai didn't know was coming, but we have Kai, Ollie, Bobby, and Pankaj who are still in this picture, so still here to date. So this was my research group at Manchester Metropolitan at the time, before I started to make the move to Newcastle in 2019, where I actually had to rebuild my lab. And then another thing shortly after that happened, which I think really shaped not just my research, but for a lot of people here, because essentially we had the COVID pandemic, and then you start to get things like this. So we had like Zoom meetings, Zoom quizzes, and then obviously there were people in Newcastle, in Manchester, so it was like a range of different things. But I think what did happen is because of this, and now everyone being familiar, first of all, I never had to explain what an antibody was anymore because people knew it because of COVID. And people became much more aware of lateral flow tests and all these other tests. So suddenly there was this whole surge of people looking at point of care sensors. And with point of care, I mean sensors that are basically here in front of you and directly give you the results. So very quick decision making. Now, obviously you will see we jumped on the bandwagon and looked at COVID-19. But even now, I still feel that there is a drive in the market to go towards these point of care sensors. Now, this is the work that we initially did in COVID-19. So we started integrating these molecular imprinted nanoparticles, working with Tesaro on detecting them, using them for detection of COVID-19. And the good thing that we could see is that at that time, first of all, they were comparable to an antibody. And at that time, there were only 2 variants. So this was quite, I wouldn't say early on in the pandemic, but at that time, there were only 2 of them. And our detection system was as good as an antibody. And also, it was capable of detecting these different variants. And that's quite important because what you do is when you do this imprinting, you can't imprint with COVID-19. No one wants to have the virus there. So what you actually need to do, and which is where it becomes far more relevant, is you need to take a very small fragment, which is still representative of the whole virus. In this case, they picked a receptor binding domain for this very selective binding. So in this case, this was probably around 12 amino acids of a whole virus. So that's a teeny tiny fragment. And we also know that there are, especially in COVID-19, there are a lot of mutants. So you need to make sure that whatever you're picking can also detect whatever is coming after this. So this was a small fragment from this receptor binding domain. And what we actually found out, if you compare it to your lateral flow tests, you might have actually noticed that it was missing cases, especially when you would start to feel like a little bit unwell, and a few days later, suddenly you would see this positive line. So actually, the lateral flow tests are not very good. And people also compare it to line eyes, which you might get for a pregnancy test, but also for the COVID lateral flow test, it was like, is this positive? Is this not positive? It doesn't really give you a very definitive answer sometimes. So luckily, we can do this with our sensor platform. And at the time, so we obviously published this also in the newspaper. And it was saying that we can get up to 6,000 times more, better in terms of the limit of detection compared to a lateral flow test. But the key thing here, and remember that I showed you this example of these lateral flow tests, which were positive because of having either Diet Coke or brandy or vodka. We could throw anything we wanted at our sensors. So basically, we could pasture it by having a very acidic or basic pH. We could heat it up, and this is what these particles look like. So first of all, you can see that the particles, this is just the diameter. So you heat it up to 50 degrees, which is basically the limit of our setup, and nothing really changes in the shape of the particles. And later on, we could also see that means that nothing was really changing in terms of the performance. Similar to the pH, we took quite a broad pH level, and you could see that the volume was really not changing that much. So in essence, we were able to measure all these things without having these false positives, or potentially without having that complicated buffer solution. We'd just take your swab sample and you could directly measure it. And this could open up opportunities, for instance, if you wanted to measure saliva or other things. And this is this comparison here where we had our test versus the commercial antigen test, so the lateral flow test. You can see it was definitely a lot better than what we anticipated. Obviously not better than PCR, but the advantage here is that you have a test where you get the result in 15 minutes, so much quicker. And then the other thing, which is what Sveta looked at, what happens if we store these sensors? How long can we keep them for? Well, in essence, a long time. So she managed to take them for over a year, and you don't need any special storage conditions, which is the other really important advantage of these polymers. You don't necessarily have to stick them in the fridge. You can just keep them at room temperature, and you can see there was hardly any change. And that's obviously like a big advantage if you look at resource limited settings, or look at very hot climates, or anything where you don't have these very specific conditions. Now, here you can see a lot more people on it, because this is where we were looking at the proof of application. So this was all good. COVID, pandemic over, no one was interested in COVID anymore. But we were able to use that knowledge to still work on virus sensing. So at the moment, as you can see, there's quite a few people looking at norovirus detection, which is now becoming very relevant again. Now having like vaccinations, there's more people than ever in hospital with norovirus. So you can see here's leafy green, so they looked at spinach samples, which maybe doesn't look as bad as the bowel samples that you will have seen, but also gave a very interesting green color. Looking at traces here on spinach samples. And then the other big thing that we looked at was looking at heart attacks. So in essence, what happens, so let's say if one of you would have like very sudden chest pain, you would go to A&E, and what people would do, they would look at the release of what a troponin, which is a protein which is released when there's damage to your heart muscle. So again, so this is a nice diagram here. We used our molecularly imprinted polymers and the thermal analysis to look and see if we could pick this up a lot quicker. Now, imagine if you are having a heart attack, time is of the essence. So first of all, you want to make sure that you pick it up soon enough. And nowadays, there's at least a 2-hour turnaround where, first of all, someone needs to take your blood. Then it actually needs to be transported to the lab. And then you need to get the results. So there's a lot of delay in it, and especially for people who actually are not experiencing heart attacks. Essentially, they're stuck in the hospital. So what we can do is, again, we present polymers are the answer to this. Well, that's what we hope. So rather than having an antibody, we have a polymer. We looked at patient samples. So we had patient samples which actually had a confirmed heart attack. And we compared, so this is our thermal technology versus the ELISA test, which is basically the standard test that we're using. So what you can see, they're not exactly 100% aligned, but you can easily correct for that offset. You can see that the trends we saw were similar. So people with high troponin levels via the standard test that had to go to the lab, which took 2 hours, versus our technology, which I said 30 minutes now, but I'm pretty confident that it can go a lot faster than that now. So huge potential for cost saving, but also for patient benefit. And I think for these examples, the nice thing of this, we used a real food sample, but also real clinical samples from the hospital. Now, finally, I want to kind of touch up a little bit about, okay, so we can replace these antibodies, but what can we do beyond antibodies? So what other functionalities can we offer? Now, what we then did, and this is the work that Pankaj started with, is Marie Curie Fellowship. We looked, in this case, at breast cancer. By the majority of breast cancer, there's an overexpression of estrogen receptor. So what we did, rather than just the traditional imprinting with one receptor, so he looked at the protein plus a drug compound. And then we postulated, and hopefully later that turned out to be true. So you have your estrogen receptor, you have your polymer selectively binding to it. Once it does, it goes into your cell, and there it releases the drug. So then what you're getting is you get that really nice selectivity, which is very important because of all the side effects you get from chemotherapy. But then the drug going directly into the nucleus of only the cancer cells, you also get a very high efficacy. So that means that you should have a highly effective treatment. And until then, this nuclear delivery was something that people hadn't seen. So this was like the first report on actually getting, with this double imprinting, getting this into the cell, because there was another paper on it out then, but that only used something which was on the outside of the cell. So now you can actually target things which are on the inside of your cell, which are much more prevalent. There's a lot of more options for using this for cancer treatment. So this kind of, you see, we have these two imprints. And looking at this, here we're kind of looking at different breast cancer types. So we say the majority of them are estrogen positive, but you also have heard of triple negative breast cancer, which is estrogen negative, which we used as a control. And these ones are wildly different. So they require very different treatment. So what we could see is once this estrogen receptor was on the cell, the material that we had was much more selective of actually recognizing those cells versus the other cells that were negative for estrogen receptor. While I say negative, in cancer you always have overexpression, so there's always a little bit of the receptor on the outside. So in essence, much more, this is your standard cell line for estrogen positive versus estrogen negative, a very big difference. And then here you can very nicely see the green is our polymer material, which had like a green tag to it. It had like the cell membrane, which is you see in red, the nucleus is in blue, so it very nicely migrated into the cell. So we could actually see that's why it was so effective. Now, once we know this, I guess there's some obvious questions that come to mind. So first of all, cancers are highly heterogeneous, and what we're using are just very simple cell lines, so it's only a 2D system. And there are so many other different types of cancers and drugs. So what we then, or what we now know, is we use some 3D model systems with a Rini value at UCL, which are more patient-like, because a real tumor has, for instance, barriers of diffusion, it has lots of other compounds that are there, it might have changes in pH. So even in a complicated 3D system, this is a polymer system, they actually show promise. But then more importantly, what we're now looking at, and this is going forward, we're looking at applying this to lung cancer with the potential of actually looking at what people do in this case in mice models, what will happen when we actually start to inject these particles. Now, what I would like to do in the future is obviously bridge these two together, so have like a sense of, and we have this real-time sensing, which you can do. This is like a standard glucose sensor working with your interstitial fluid, so real-time monitoring. So having something which then tells you via AI, okay, this might be off, you need some medication, but then also implementing this kind of reactive drug delivery in the process. So hopefully this is what I'm hoping to do in the future, bring all of this together and then using the technology that I have to develop new tools. Now, this will be very brief, but what I also wanted to highlight is now we've done all the academic things, but obviously, as we know in academia, very little of what we actually do has real life impact. So we are actively working with companies, so hopefully we use the kind of these companies to actually build on this and to achieve this impact. And what Kai started doing towards the end of the PhD is looking at different ways of how we could miniaturize the system. And the way to do that, we thought at the time, so we have like what we call these thermocouples, which are different ways of how you measure the temperature. They had like these little wires. We had a system which is around 5 kilograms, so we classed it as portable in our publications, but it really wasn't portable. And we worked towards like small little chips, so these are like little beads from misters, which are actually, your thermocouples are much more robust, so that's why they're widely used in industry. They're really good at measuring like a wide range. However, thermistors, they work in a very narrow range. So this is the signal you're getting from your thermistor, so much cleaner compared to the thermocouples. So what we were thinking is, what if we just replace them? And luckily for us, that worked out okay. So we published this first. This was actually a while ago. This was still in Manchester Met. And now recently we started coming back to this, thinking like, okay, how could we develop a new setup from this? Because luckily these thermistors are much cheaper, so you actually have potential for single use. So for instance, thinking for this COVID test, you would have like your sensor, which is disposable, but also like a little measurement cell, which you can just throw away. Now, this is the comparison 1-to-1. So I said this was £1,500 we had in the beginning to like 10 times cheaper, much more compact. But nowadays it looks like, I think I didn't have the example of the little suitcase in there, which we use it in now. But essentially it looks a little bit, these are kind of electrodes, which are literally kind of this size. You would slot them in here, and the way hopefully it would work, think of it like an espresso system. You want to detect one thing, you have a particular pod. You want another flavor, you add a different pod. So you want to detect another sensor, different pod, and so on and so on. So this is hopefully what we're kind of on this journey now via the iCare journey with Alexander leading this, but a lot of other people being involved, saying, what can we do with this kind of customizable sensors? And for now, we're going to focus on heart attacks, but what we also found in the journey, there's definitely demand from like other sectors to look at this type of technology. Now, hopefully, I try and pitch this with giving like a few takeaway messages. So I think that these designer polymers will find their applications in healthcare. They're not widely implemented yet, and until now I think a lot of people here in the room might have not heard of these molecular imprinted polymers, but because of them being highly versatile, robust, and more importantly cost effective, I think these will become really important. So now the antibody therapies, for instance, are very good at treating cancer, but they're definitely not accessible to the majority of the people, even here in the UK, or definitely not in the US or other countries. So we rapidly need other solutions for this. Now, unfortunately, the only commercial application so far on these molecular imprinted polymers is for separation, where they're using these like microparticles, which is what I used in the beginning of my PhD, which is definitely not the best route to go down to. So what I think will happen is that we just need to find one killer application where you say like, okay, why can we just, because now people are like, why don't we use antibodies? You need one really good example to show why this is important. Now, in the future, we can't get around it anymore. Like, modeling has never been my favorite. But if you could automate the whole process from the way to the design, to the manufacturing, to the analysis, and using AI and modeling, and this is what we're proposing now, I think this is where the future is. Because, in essence, if you have your polymers, you have an infinite number of building blocks to work with. So why shouldn't we be better than an antibody which is only made out of 20 amino acids? Well, if it was that easy, people would have done it. But this is kind of what it then would look like, where you have high throughput screening, which you need rational design using modeling, synthesis, and machine learning. Now, a few other small things, which is more like, I guess, on a personal note. So what I think I really enjoyed throughout my career is this multidisciplinary working and investing in the people that I've worked with. So there's a lot of people here in the room that I've worked with for a very long time. So it's really good to kind of see them coming back here, and hopefully they will see some of the impact they've had or where we are to date. And what I've also really enjoyed, and which is not necessarily what academics tend to do, I've done quite a lot of public outreach, but I think that's really kind of helped to explain the problems, hopefully in an understandable manner, but also engaging with companies, etc., in order to see where we're going with this research. Because I think only if you kind of have that curiosity to see what's next, then this is where you're getting, I think, the next breakthroughs for your research. And so I've engaged a lot with, I have lots of videos on YouTube, but also with the Royal Society of Chemistry, and particularly with IUPAC, where I met a lot of other polymer scientists who've really helped me along the way. Now, these are just some of the, I guess, like scientific collaborations. So obviously, I know this is not everyone. I tried to make a slide, so I tried to do it per project. But I know some people are across different projects. These are some of our funders. But there's a lot. So this is mainly the people who are previous and current PDRAs, PhD students who've done a lot of actually the hard experimental work. And this doesn't even include the 20 Brazilian students I've supervised with Professor Jonas Kruger, which is a big thing. And what I'm also really fortunate with, that some of the people, like for instance my PhD supervisor, have a continuing collaboration with, and some people are in the room, like Alex from Industrial Input, and also Mark and Nick are here. So that's very nice to see them coming together to see the work that we're doing.